So, good morning, dear students. I welcome you for today's first session, and indeed, it's my pleasure and privilege to introduce our colleagues and friends, Dr. Niraj Singh, who is currently working as a associate professor, head Department of Biotechnology, Genetics and Bioinformatics, Sri N. Patel College of Pure, Sri N. Patel PG Institute of Science, Research, Anand. So far, Dr. Niraj sir is concerned, he is one of the good academician and researcher. He has very vast research and teaching experience since 14 years, and he himself is a product of various competitive entrance examination. He cleared JNU entrance examination and did PG from University of Pune. Thereafter, he cleared CSIR NET, ICMR NET, ICR NET, GATE, everything, whatever is needed for PhD program. He did PhD from Sardar Patel University. He has published more than 20 research papers in different international and national journal of repute, having good ranking. He completed several research projects including DPT, DST, GSBTM. He got guideship from Sardar Patel University, and presently 2 students are pursuing PhD under his supervision. And he has very vast experience of teaching so far entrance examination is concerned. He is member of Gujarat State Biotechnology Mission PG CBC. He is member of Network for Capacity Building Set UG for PG entrance examination. These are the brief and abstract about his achievement. Without taking and wasting much time, I welcome you behalf of GSBTM and NBT CBC NB Pass sir, and I request Niraj sir to please proceed further. Sir, please.
Thank you very much, Kundan sir. So, good morning, friends. So, today we discuss classical genetics, and when we start lecture, so we start lecture from a question that why the children in a family resembles to one another, but they are not resembling exactly, right? So they resemble to some extent, not exactly. So this can be answered by a branch of science, or this question can be answered by a branch of science that is called genetics, right? So first of all, we define the genetics. So genetics is nothing but the branch of biology which deals with the heredity and variation, right? So you always keep in these two terms in your mind throughout the lecture, right? So one term is heredity and another is variation, and genetics is the science which deals with the heredity as well as the variation, right? So first of all, we try to understand what is heredity and what is the variation. These two terms are exactly opposite term to each other. Heredity is nothing but the factors which are responsible for the resemblance between the parents and the offspring, right? Whereas the variation, it is the force which influences the two organisms to not exactly alike, right? So they are different actually, right? So these two terms are exactly opposite to each other. So first of all, we discuss the historical background of the heredity. So this concept of heredity is not new. Actually, this is very old. Our farmers, they were doing the selective breeding of their horses, donkeys, trees long back in the ancient civilization, 6,000 to 8,000 years back. They were doing selective breeding, right? So this science is not new actually. This is very old science, but this, selective breeding and other aspect has been studied scientifically by a scientist known as Gregor John Mendel in 1866, At that time, his work was not recognized very well, but in 1900, their work, Mendel's work, were re-established, or we can say the rediscovered, and then people came to know the importance of Mendel's work. So the brief scientific contribution to the genetics is like that. So first, the Gregor John Mendel in 1865 approximately, right, did many work related to the inheritance and the variation, and Mendel is called father of genetics. He gave the concept which is called factor, concept of factor. Today we call this factor as a gene, right? But at that time there were, no such sophisticated technique and nothing over there. So Mendel hypothesized a hypothetical factor, and these hypothetical factor Mendel considered as the unit of heredity. And almost at the same time in 1869, Friedrich Mischer, he discovered nucleic acid from the pus cells, right? So pus cells, he discovered nucleic acid. And in 1900, 3 scientists from the different countries, Karl Korens, Hugo de Vries and Cermak, he rediscovered the Mendelism, right? Then in 1902, Boveri and Sutton, they identified a very, very important thing, that those factors, what Mendel hypothesized, and the gene, what we call today, they are located on the chromosome, right? Then in 1906, the Batson, he gave the term genetics and was proposed the study of heredity. This is nothing but the branch of science which deals with the heredity, right? And he also gave a term which is called allele or allelomorph, right? We discuss these terms in the next slides. Then Johansen, he gave a term genes for the factor, Mendelian factor. Then 1928, very, very important work done by the Frederick Griffith, right? And Frederick Griffith, he performed a very elegant experiment called transformation experiment by taking Streptococcus pneumoniae, and he tried to establish the factors or gene or the thing which is responsible for the heredity and the variation. He awarded with the Nobel Prize also, but he was not succeeded to, he was not successful to, call very strongly that the DNA genetic material. So his student, 3 students, Avery, MacLeod and McCarthy, right? They are student of the Frederick Griffith, and they established that DNA is genetic material, right? But again, they were not able to explain why the proteins are not acts as genetic material, because whenever you are isolating DNA, so proteins are always with them, right? So that question has not been answered by the Avery, MacLeod and McCarthy, right? They also awarded with Nobel Prize, but that question was answered in 1952 by Hershey and Chase, right? And they finally proved proteins are not genetic material, only the DNA is genetic material. Now it is established, and then the modern the molecular biology comes after the discovery of the Watson and Crick. So first of all, before discussing the classical genetics, we just discuss few terms. So first is the allele, right? So allele is nothing but the alternative form of the gene, right? Alternative form of a gene, right? Like, for the height, a gene which can be named as T is responsible for the height of a plant. So we know very well in eukaryotes, 2 sets of chromosomes are there, and on these 2 sets genes are located, and those genes are located exactly in front of each other, right? So one gene is T from the maternal side, suppose, and this gene, I mean this, gene is from the paternal side. This is from the maternal side and this is from the paternal side. So these two alternative forms of a gene are called allele, right? So this is one allele, this is another allele, or it is also called allelomorph. Okay. Second is homozygous. If these alleles are present in the same form, right? So they are called homozygous, either in this form or in this form. This is called homozygous. And if they are present, in two different alternative forms, then it is called heterozygous, right? Capital T, small t, right? Hemizygous is another term, which is nothing but many a times what happens, only one allele is present in many cases, like in human males, right? In human males, if we talk about the sex chromosome, right? So XY is there, right? So the X chromosome, structure of the X chromosome and the Y chromosome is very different from each other. So genes in front of on the X is not exactly onto the Y, or they are not, alternative form of each other. So the genes present here on X, suppose for the eye color, the gene, suppose E is there. So that, or not actually, we take another example. Suppose for the testis, right? So testis determining factor, right? TDF, gene for the TDF, suppose present on the Y, right? So this is the T, so that gene is not present on the X here, right? So we can present like this, the gene for T is there, but gene on the X is not present. So in this condition it is called hemizygous. So only one form of the gene is there, so that case is called hemizygous, right? So first of all we discuss the Mendel's work. So the Mendel, he worked on garden pea called Pisum sativum, and he tried to understand the inheritance pattern, how the inheritance or how the variation takes place in the pea. So he took many characters to study the Pisum sativum, and it took almost more than 7 years to study. And after the study he proposed law of inheritance, right? So the Mendel was very successful. Mendel was very successful to propose the principles of the inheritance. Many reasons are there. One of the very strong reason is that Mendel first time used the statistical analysis, or he used mathematical logic to solve the problems which are there in the inheritance biology, right? Another very important thing for the success of Mendel was the large sample size, very large sample size he studied. Then the confirmation of his inference to many generation, actually. He did many more generation for the testing, right? To study the inheritance pattern, to understand the inheritance pattern, right? And for his investigation, Mendel took two opposite traits every time, right? That is called contrasting characters, right? Like, tall plant, he took tall plant and crossed with the dwarf plant. Another like yellow plant and crossed with the green seeds, yellow seeds and green seeds, right? So two contrasting character he took every time. Okay, so these are the following regions for the success of Mendel. So quickly we go through this slide. So the pea plant is very easy to cultivate in the open ground. The flowers of pea plant are normally self-fertilized, right? But the cross fertilization can also be done very easily, right? So another thing, the number of contrasting characters are very high in pea plants. Then the hybrid pea are also fertile, right? So cross pollination is also can be done, is not very difficult in the pea plant. Then the artificial fertilization, that is cross pollination or fertilization, is almost successful. Then the very important thing about Mendel is that he was very patient actually. So he studied only one character at a time. So this is a very important point why Mendel is so successful among the other scientists. So he took only one characters at a time in most of the experiments, and after understanding, then he took more than one characters for the study. So another very important thing is that he maintained the statistical record, right? And that mathematical or statistical record, it helped Mendel to drive the mathematical ratios and the significance. Okay. So before starting the experiment, Mendel make true breed of the pea lines. Suppose he took tall plants. So now we can understand for the tall plant, two genotypes are possible: capital T and capital T small t, right? But at that time it was totally unaware that so two are nothing. But he always tried to make the capital T genotype for the parental generation, right? Because for the tall plant it is very difficult to say whether it is capital T capital form or capital T small t form. But Mendel was really very intelligent. So what he did, actually, he did selfing for many generations. Suppose a plant which is having genotype capital T, so he's made a selfing among the capital T. So at the end, what he will get? He will get 4 different kind of plants, right? 4 different plants having. Mendel was not aware of these genotypes. Always keep in mind, right? So this is capital T small t. This is also capital T small t, and this is small t. So 3 type of plants are there, right? So this is small t plant. So this is dwarf, right? So this is always dwarf plant or the recessive, characters which shown by the recessive traits are always in the true breeding line. So this plant, this dwarf plant is always in the true breeding form. But here these plants are the tall plant, right? So again, what he did, Mendel, he made selfing of this capital T, selfing of this capital T small t. So what he get in the next generation by having this plant, this capital T selfing, so he will always get capital T all tall he will get, right? But here the tall plants as well as the dwarf plant will get, right? So it means that these capital T small t are not true breed, right? So those plants which gives always dominant characters are considered as true breed, right? So Mendel first of all selected the true breeding line. He made the true breeding line, right? And then he started his experiment, right? So in that way, Mendel made 14 true breeding pea plants varieties, right? And those are considered as the parental plants, pea plant, right? P, we provide a notation for the parental generation is P, right? And then the parental plants like capital T, this is true breed, right? And the another true breed which is dwarf plant, small t, right? He took as a parental generation, parental plant, and then he made cross between the parental generation and he got the generation in the of the genotype capital T small t. So this is called F1 generation, right? First filial generation, right? And then after selfing the F1 generation, then he got F2 generation, right? And after selfing the F2 generation, he got F3 generation. So F1, F2, F3, first filial generation, second filial generation, third filial generations are there likewise. So this slide is very, very important for your exam point of view, right? So the 7 characters, 7 traits which Mendel was selected is given here. right? The dominant character is here and the recessive characters are here in this panel, and these are the characters shown by the F1 hybrid, right? Okay, all right. So you try to remember these different dominant as well as the recessive character. Many a times they are asking, right? Like round seed is dominant over the wrinkle seed, the yellow seed color is dominant over the green seed color, then purple flower is dominant over the white flower, etc., etc. Here tall is, or standard we can say here, the tall is dominant over the dwarf. Okay. So in the monohybrid experiment, so Mendel did many experiment by taking one characters, they call them monohybrid cross, right? So single gene inheritance, so because they took one character at a time, right? And he took tall plants, I mean true breed tall plant, right? Always you understand true breed, right? Here in the parental generation, and the dwarf pea plant, right? And then he studied the F1 generation, right? So in the F1 generation he got a very, very specific ratio, right? Some plant, I mean all plants are tall plants in the F1 generation, right? And then he made a selfing of the F1 generation and he got F2 generation. So for the crossing, what Mendel did, Mendel took two different plants, like this is one plant having violet flower and this is white flower, suppose. So because the both sexes are present on in the flower, so he removed the anthers, that is emasculation. So from one flower he removed the anthers, right? And from the second flower he took the the pollen grains, he dusted onto the first flower, right? So in this way they made a cross pollination arrangement, right? So emasculation and then the dusting of the pollen grains onto the emasculated flowers, and he got all tall plants in the F1 generation, right? So Mendel got 1064 plants, right? And out of them in the F2 generation actually, so out of them 787 plants are tall and 277 plants are dwarf. So he got a ratio of 3:1, phenotypic ratio, right? So 3:1, right? So these three plants are tall plant, right? And one plant is dwarf plant, right? So the monohybrid cross and the phenotypic ratio is 3:1, right? See, the important thing about the monohybrid cross is that the contrasting traits, I mean the trait for the tallness and the dwarfness did not show any blending, right? Because both the traits are here, I mean both the alleles are here, capital T and small t, but both the traits are separated from each other during the gametogenesis or gamete formation, and finally they form the dwarf plant also. So we can say here the traits are not blended either in the F1 generation or F2 generation. As and when required, they separated from each other. Okay. So there is a question in 2018 JNU entrance examination. Just this is the probability question actually. So in animals, the inability to make the melanin results in the albinism, right? And for the albinism, the genotype would be small a, small a. So recessive, if both the genes are there in the recessive homozygous form, so albinism is there. So the question is, what is the probability that the second and third children will also albino, right? And the unaffected parents, the parents are unaffected. So we can draw the genotype of the parent. So both the parents are unaffected, right? So if we take the genotype of mother, that should be capital A, small a, isn't it? Because the mother is unaffected, father is also unaffected, right? And then here two different kind of gametes can be made, capital A and here is small a, Here also two different type of gametes can be formed, capital A, small a, right? And after crossing, so if we cross them, then this, this is capital A, this is normal, right? So this is capital A, small a, this is also normal, right? This is small a, capital A, this is also normal, right? But here this is small a, this is albino, right? So the question is, what is the chances, what is the chance to get the albino? So here the 1/2, right, probability of making the recessive gamete is half, and here also it is half, right? So the half and half probability is there of making the albino, so it would be 1/4. So 1/4 is the probability of making the albino child, right? So answer would be the 1/4, right? So based on the monohybrid cross, Mendel, they proposed two general rules actually, right? The first rule was the law of dominance, and second rule was the law of segregation, right? So later these general rules were called principles of law of inheritance, right? So these two rules are based on the monohybrid cross, keep in mind, right? Monohybrid cross, right? Okay. So law of dominance is nothing. The out of two allele, the only one allele is expressed itself if they are present in the heterozygous condition. Suppose the F1 generation, the genotype is capital T, small t, so only capital T is expressed, not the small t, right? So this is called law of dominance. So here the small t, the expression of this gene is not there at all, always keep in mind, right? So other allele remains unexpressed, right? So here the if you examine, then you will find the, so after getting, I'm sorry, just a minute. Okay, so this is the F1 generation. So this is tall plant, this is dwarf plant, right? So here all tall will be there, right? All tall, right? Capital T, small t genotype, and after selfing, the two different kind of plant, or morphologically, phenotypically two different kind of plant. One is the tall population and another is the dwarf population is there, right? So in the tall, three plants are there, right? So here the phenotypic ratio is 3:1, right? And the genotypic ratio is, this is one genotype capital T, and two genotype, that is capital T, small t, that is heterozygous, and one is homozygous recessive. So the ratio is 1:2. So genotypic ratio is 1:2:1. Okay. So another observation or another general rule proposed by Mendel is the law of segregation. So law of segregation is nothing but what it tells, that two alleles, capital T and small t, suppose, right? So these two alleles remain together without any mixing of the characters, right? Suppose in the F1 generation, the plants are capital T, small t, right? So they remain together, capital T and small t, in the F1 generation, but these two alleles remain there without any mixing, right? But at the time of gamete formation, they separate from each other without affecting to the another allele, right? So this is called law of segregation, this law, and this law is also called law of purity of gamete. So gametes are always pure, right? Gametes are always pure because only one allele is there in the gamete, right? That's why this law is also called law of purity of gamete. So these two laws were proposed by the study of the monohybrid cross, right? So now the question is that Mendel, he was intelligent, as I mentioned earlier, but how he came to know the genotype of the F1 generation, right, was very interesting, is very interesting actually. So no such device is there to find out or to see the homozygosity or heterozygosity or the genotype of the F1 generation. So Mendel, he devised a tool which is called backcross, right? So backcross is nothing but he made for the analysis of the F1 genotype, whether it is homozygous or heterozygous, right? So two different kind of tests he did. When the F1 individual cross with the dominant parent, suppose F1 individual, this is capital T small t is the F1 individual. When he crossed this individual with the dominant one, right? So in that case, he will get all tall, isn't it? So, I mean, all tall, the genotype may be different, capital T and capital T small t, two different kind of genotype has been made here, but phenotypically all the plants are tall, right? So this is one experiment, and this is called outcross, right? This backcross is called outcross, right? When the F1 individual crossed with the dominant parent. So no analysis is possible in the F1 generation, right? By having the outcross, because always the tall plant has been made, right? Another cross he devised, that is called test cross, and test cross is nothing but the F1 individual when crossed with the recessive one, right? Then the scenario is different. Some plants are tall, I mean capital T small t genotype would be there, right? And for others it would be small t and small t, right? Because here the gamete is only small t, right? Here the capital T small t is there, and two different kind of gametes are there, and if you cross them, right, if you cross them, this capital T and this is small t, right, and this is small t and this is small t, right? So two different kind of plant, or morphologically Mendel can distinguish them. So by having this, you know, the F2 generation or the test cross result, Mendel can conclude, or Mendel came to understand, that F1 plants are always heterozygous actually, right? When he took, because he took true breed of capital T and dwarf small t and small t, right? So by having the test cross, he came to know the F1 generation or F1 plants are actually heterozygous. So one can establish the genotype of the F1 generation by having the test cross or outcross, or we can call the back cross. So here they can ask you, so test cross may be a back cross. So yes, this is correct, but all back cross are test cross, so no. So all test cross can be called back cross, but all back cross cannot be called test cross, right? All out cross can be called back cross, but all back cross cannot be called out cross, isn't it? So this is the, you know, tool by which one can find out the genotype, and particularly the F1 genotype, right? Here this is the diagram. Suppose the genotype of this flower is not known. This may be capital W, violet flower is there, and capital W, small w. So when you cross them here by the recessive parent, this is the recessive parent, right? Both here this is the first experiment, and this side this is second experiment, right? So when it is crossed with the white flower, right? So if 50-50% ratio, right? This is violet flower and this is also white flower. So this ratio is 1:1. So if it is 1:1, then it means the genotype of your test plant is in the heterozygous form, right? But if it is all violet, no ratio is obtained, all dominant form it will express, then the genotype would be homozygous dominant, not heterozygous, right? So the genotype of a plant or a flower can be established by making the test cross, So test cross is a very important tool, right? So now if you got those things, then you can be able to solve this question. So which of the following cross would produce a 1:1 ratio of phenotype in the next generation? So certainly it would be test cross. So test cross is nothing but the F1 generation when crossed with the homozygous recessive parent, right? So the answer would be, this would be the correct answer, right? D would be the correct answer. Okay. You can solve this question also. This is in the IIT JM 2019. So when the yellow color Y is completely dominant over the Y, small y, the genetic cross between the plant with yellow seeds and plant with the white seed yield a progeny of 48 yellow seeds, right? 48 yellow seed, right? So yellow is dominant, and 52 white seed, this is recessive, right? The genotype of the yellow seed and the white seed parent plant respectively are most likely to be. See, okay, so this is 48 and this is 52. So ratio is almost 1:1. This is not exactly 1:1, right? But this is almost 1:1. So they are asking, they are trying to ask in which cross the ratio would be 1:1. So of course this is the test cross, right? So in the test cross the ratio would be 1:1. So we have to find out the test cross. So this would be the situation where test cross is given, right? Okay. See, this is the monohybrid test cross ratio. Always you keep in the mind. This is what we got, monohybrid cross, right? Monohybrid we are getting 1:1, but in the dihybrid test cross, this would be 1:1:1:1, right? I mean, two traits if taken and then you perform a test cross, then you will get 1:1:1:1 ratio. All right. So test cross, many questions they are asking. Almost every year they are asking questions. Okay. So now after getting sufficient information, when the Mendel understood very clearly the pattern of inheritance by monohybrid cross, then he started doing dihybrid cross or dihybrid experiment, right? So this is nothing but the inheritance of two genes, right? So two genes are taken at a time, right? Like shape of the seed and color of the seed, right? Two traits are taken together, right? And again he made a true breed. Always keep in mind, before starting the experiment, Mendel made a true breed. So parental plant he made and he crossed the dominant as well as the recessive true breeding plant, right? And he got a ratio like this. So when he took round and yellow seed, this is RRYY, right? And this is recessive, wrinkle and green. So in the F1 generation, the RR, capital R small r and capital Y small y seed will get, right? And after selfing, he got this kind of, we can make a trial here also. So see, the four different kind of gametes they are forming, right? So we can take here, male plant RR and YY, and here also RR and YY, right? Four different kind of gametes here they produce, right? So this is RY, this is R small y, this is R capital Y, and this is RY, right? Here also same kind of gamete is there. So all gametes are written here, male gametes here and the female gametes here. And when you make a cross, then you get this, suppose this RY, and here from here also this RY. So this will make a genotype of this capital R, capital Y, capital Y, right? So you will get a ratio or the phenotypic ratio which is 9:3:3:1, right? 9 plants are round and yellow right? Round and yellow. See, these 9 plants are round and yellow. Their genotype may be different. We come to that point also, right? Then the 3 plants, which is wrinkle and yellow. See, this plant wrinkle and yellow, this is wrinkle and yellow, right? And 3 plants round and green, right? Round and green, and 1 plant is wrinkle and green is there. So this ratio is, the phenotypic ratio is 9:3:3:1, right? But when we check its genotypic ratio, right? So genotypic ratio will be a bit different, right? So just you see and write it down. So when you write it down, and this can be written very easily, and you can remember this genotypic form: 1 2 1 2 4 2, right? 2 4 2, and then again this is 1 2 1, right? Just you find out the genotype. So one RR, capital R, capital R, capital Y, capital Y. So this may be this one, right? Another, the small r, small r, small y, small y. So this would be the ratio. So you always keep in mind, right? Just you can match this with the table, this Punnett square diagram, right? So 121, 242 and 121, right? दोनों तरफ 121, 121 और बीच में 242 को रख देना है, right? So this is the genotypic ratio of the F1 cross, right? Monohybrid में कितना था? Monohybrid में it was 1:2:1. That was the genotypic ratio, right? So here what happens actually? The earlier the cross was made like this. What arrow we are making here, like this, and then like this. But Punnett, he was a scientist, and he just made these crosses in this square presentation form, right? That's why this square presentation form is called Punnett square diagram, right? So this is called Punnett square diagram, all right? Okay, so this is genotypic as well as the phenotypic ratio of the dihybrid cross, right? So after the study of the dihybrid cross, Mendel proposed third law, and that law is called law of independent assortment, right? So what he observed these two traits, that means yellow and not yellow, the seed shape and the seed color, right? These two traits he has been taken simultaneously, but he observed these two characters are not interfering each other, right? They are separated in the next generation without interference. They are assorted independently without interfering to each other. So he proposed third law and called law of independent assortment. See the difference between the law of segregation, right? The second law was law of segregation. This is the third law, right? So law of segregation was nothing but he was talking about the separation of allele, right? So no interference between the two alleles are there, even though they are in the same cell, right? They are together without interference, right? During the gamete formation they separate from each other without interference. But here in the third law, law of independent assortment, they are talking about two traits, right? Two traits, two different characters, okay? Like shape, right? Shape and color, right? So two different characters are separated from each other in the next generation without interference. So this is called law of independent assortment. See, three laws, all the three laws. Now we just concluding these three laws. So first law, law of dominance, this is true, but this is not always true. In all the organisms, this is not true. After Mendel's work, many scientists worked, right? And they found this law is not always true. The second law, law of segregation. So this, or law of purity of gamete, this law is always true, right? Always keep in mind. And third law, law of independent assortment, is also not true in all cases, right? So they ask you in the exam, which of the following law of the Mendel is valid till date, or it is applicable always, everywhere? So that is nothing but the second law, right? And that was the law of purity of gamete, or law of segregation, right? Okay. So the evaluation, okay, so this is nothing but the, after the Mendel's work, many more scientists, they work by taking the Mendel's pattern, and they are getting bit different ratios, right? They were not getting the ratios which Mendel was getting. So Mendel, he was getting 3:1 ratio in monohybrid cross phenotypic ratio, and 9:3:3:1 phenotypic ratio. So these ratios are not getting in the different organisms by different group of scientists. So those are called post-Mendelian work, right? So now we discuss post-Mendelian work, right? But before discussing the post-Mendelian work, these are the very important shortcuts, and certainly you would be very happy to solve the questions in a very few seconds actually, by keeping these formulas in the mind. Suppose someone is asking in the question that how many type of gametes are formed, like here. This is very simple actually, capital T, small t, so two different kind of gametes are formed, right? But if I'm asking you, if this is the genotype, right? If this is the genotype, capital C and capital D, then it is a bit difficult to tell, within a second that how many gametes are formed by sketching the number of gametes. So you keep one formula in your mind, 2 raised to the power n, right? So this is nothing but the n is the number of heterozygosity. So you always find out the heterozygosity, how many heterozygosity is there. Like here, 1 heterozygosity is there, right? So 2 raised to the power 1 means 2. I'm sorry, 2 raised to the power 1 means 2, yeah. Okay, so only 2 different kind of gametes can be formed. Another example I'm giving you. Suppose capital T is there, then there is no heterozygosity. इसमें heterozygosity है ही नहीं, right? So 2 raised to the power 0. So 2 raised to the power 0 is 1. So only one kind of gamete has been formed. Now you come to this big question here. So now first you find out the heterozygosity. So this is homozygous. This is heterozygous 1. This is homozygous. This is again heterozygous. So 2. So number of heterozygosity is 2. So what you do? 2 raised to the power 2 is equals to 4. So number of gamete formed is 4, right? And the phenotypic category is also calculated by this formula only. How many phenotypes would be there? Like see here, the capital, suppose F1 generation, you are making a self cross or self pollination of the F1 generation, capital T. So how many phenotype would be there? So 3:1, 3 tall plant, 1 dwarf plant, right? So 2 phenotypes are there. So same formula, 2 raised to the power 1, heterozygosity is 1. So 2 phenotypes are there, right? So this is very important and useful formula. Always keep in mind. Okay. So type of genotype, how many genotypes are there? So 3 is to n, 3 raised to power n. So again, n is the heterozygosity. So you just take one example. Suppose monohybrid cross, capital T, small t. So जीनोटाइप कितना मिला था वहां पे हमें? तो 3 जीनोटाइप मिला था. कैसे? जब सेल्फिंग किया था किसका? F1 जनरेशन का. तो कैपिटल टी दूसरा मिला था, कैपिटल टी स्मॉल टी, एंड थर्ड वाज द स्मॉल टी. ऑलराइट. सो 3. तो फॉर्मूला यहाँ पे रख दो. 3 raised to the power 1. Heterozygosity is 1. So this is 3. Alright. So 3 genotypes has been made. Phenotype करना हो तो 2 raised to the power n. Genotype करना हो तो 3 raised to the power n. Okay. The number of zygote, third formula is number of zygote produced by the selfing. कितने zygote बनेंगे, right? तो इसको कर देना 4 raised to the power n. जैसे आप देख लो यहाँ पे हेट्रोजाइगोसिटी, यहाँ पे F1 क्रॉस में हमने देखा कि जाइगोट कितने बनेंगे, राइट? F1 क्रॉस में जब जाइगोट बनाओगे तो ये 4 जाइगोट्स आर देयर है ना. सो दिस कैपिटल T स्मॉल t आर 2 in number, isn't it? So this is 4 raised to the power 1. 1 means heterozygosity. This is heterozygosity, and so 4 raised to the power 1 is equals to 4, right? Again, you can check this formula in the dihybrid cross also. So dihybrid cross in the F2 generation, F2 generation, what happens? See, 16, just you 9 plus 3 plus 3 plus 1, total 16, different zygotes are there. So number of heterozygosity in dihybrid cross is 2. Suppose R, R, Y, Y, right? So heterozygosity कितना हुआ इसमें? 2. So what you do? 4 raised to the power 2. 4 ja 16. So 16 zygotes are formed. So these all three formulas are very, very important and useful formula for you. Okay. Okay, now we discuss the post-Mendelian work. So in the post-Mendelian work, what I mentioned earlier, the law of dominance was not true actually, always, right? It was true, it is true in fact, but it is not always true. So again, the law of independent assortment, that is also not true for all the cases. That was observed by the many group of scientists later to the Mendel's work, right? So what they found out, the gene interaction is there in many cases, right? So we just try to understand what is the gene interaction. So the allelic interaction, it is of two types, allelic interaction and the non-allelic interaction, right? So allelic interaction, first we discuss. So the first is the incomplete dominance, right? So incomplete dominance has been observed in the Mirabilis jalapa. Then that is 4 o'clock plant, Antirrhinum majus, dog flower, right? So these are the plants where the incomplete dominance has been observed. So this is something like this, the red flower when crossed with the ivory flower. So pink flower has been obtained in the F1 generation, right? So what happens here actually, the mixing of the red color, right, as well as the white color, it gives pink color. So both gene express, right? So the capital I and small I both will express, right, and they mix together and give the new phenotype. So this is called incomplete dominance, right? So in the incomplete dominance, if we look at the ratio, right, so ratio would be, this is the red, pure red, I mean capital I, this is pink, this is white or ivory. So the ratio is 1:2:1. So both genotypic as well as the phenotypic ratio is 1:2:1. Another is the codominance. Codominance that is seen in the fur color of the many animals, right? ABO blood grouping we can see, right? The codominance. What happens codominance actually? Both gene, they express themselves, right, but their products are not intermixed, right? So here the capital R it gives red, but capital R small r it gives reddish as well as the gray color, right, whereas the R is the white color, right? So the coat color of the cattles, they kept into the incomplete dominance, right? Sorry, codominance. So the incomplete and the codominance, the difference is that in incomplete dominance, the gene product are intermixed, always keep in mind, right? And it gives a new phenotype. Here, the both gene products are not intermixed, right? They are independently showing their phenotype, right? So again, the ratio here also is 1:2:1, okay? ABO blood grouping is another example, right? So in the ABO blood grouping, so the antigens on the RBC, here the IA, the antigen is the N-acetyl glucosamine, right? On the B, IB, it is galactose, right? This is the precursor carbohydrate, right? So this can react with the antibody A, and this can be react with the antibody B, right? So here in the case of AB blood grouping, right, AB blood group की बात करें तो here the genotype is IA and IB, so both are expressing equally, and example of codominance, right? Third is sickle cell anemia. So in the sickle cell anemia, here the HBS and HBS, right, HBA and HBS, right, heterozygous condition, they are expressing equally, the products are not intermixed. So some RBC are sickle shape, and this is due to a point mutation at the 6th position of the beta chain, where the glutamic acid is replaced by the valine, right? So always keep in mind. So this is also an example of codominance. Okay. Third is the multiple allelism. So multiple allelism, what happens? What Mendel said, the only one allele, or the, for a trait, two alleles are there, right? Capital T and small t. But what was observed later, many more alleles may be responsible for a single character, like blood grouping. Three different alleles are there, IA, IB and small I, right? Three alleles are there. So blood grouping is an example of dominance also. See, dominance, like blood group A, IA, small I, so this is blood group A. Then for AB, this is capital A, this is the example of codominance, right? And multiple allelism, this is an example of multiple allelism also, because multiple alleles are required for a single kind of trait in a population, right? Another is pleiotropism. So pleiotropism is nothing but a single gene have more than one characters or more than one trait they can regulate, right? So this is some examples are sickle cell anemia, sickle cell disease, the pigmentation in the cat or mouse, right? So these are the example of pleiotropism. So now the second one is the non-allelic interaction. That was the allelic interaction. So now we discuss the non-allelic interaction. So non-allelic interaction means the two different genes, interaction between the two different genes are there, right? So suppose a gene X is there which produces some substance that is Y, right, by the action of enzyme 1, and in the reaction 2, the enzyme 2, it produces a red flower, right? So here from white flower, the formation of red flower, two reactions has been required, right? So two genes are involved, capital R, capital C. So these interactions, when you study, it gives the deviation from the 9331 ratio, and this can be categorized into complementary gene, inhibitory gene, epistasis, duplicate gene and additive gene. We discuss one by one. So first is incomplete or complementary genes. So in the complementary gene, just to try to understand the pathway, complement, I mean one gene will complement the another. If enzyme 1 and enzyme 2 both are present, then it is red flower, right? If any one of them are missing, then they won't produce red flower. That means white flower is there. So here, see, the capital R and small c. So second enzyme is not there. So flower is white, right? Second here, the small r. So first enzyme is not there. So again the flower is white. Here both enzymes are absent. So flower color is white. But here both the genes are there. Both the capital R, capital C. So both enzymes are there. So flower color is red. So if you look at the ratio, I mean genotypic ratio, I mean what is the altered ratio from the 9331? So see here, only this flower is red flower, and remaining these three flowers are the white flowers. Okay, so this is 9 red flower and 7 white flowers are there. So here complementary gene, the ratio becomes 9:7. So this is a kind of gene interaction, right? Second, inhibitory gene. So inhibitory gene, what happens when the enzyme 2 is there? Same pathway is there in almost all the 5 interaction. So if enzyme 2 is there, so again it becomes white flower. So it started from white, the enzyme 1 is present, it gives red color, but when enzyme 2 is there, it inhibits the red color to again white color, right? Okay. So we directly come on to the ratio. So see here, if only the capital R is there and the another is small I, then only it is red, otherwise all the combinations are white. Okay, see here in the 9, capital R and capital I would be there, so flower color would be white, right? And only the 3 flowers are there in which only one capital R is there, only capital R is there, so it is red, right? Here the small r, here also small r, so it would be white. So this is 9 these two are 6, so 9 plus 6, 13. How much? 9 plus 4, I'm sorry, this is 4, right? So 9 plus 4, 13, and this is red. So the ratio becomes 13:3, right? So in the inhibitory gene, the ratio would be 13:3. The third is the epistasis. You try to understand. So here the enzyme 1 and enzyme 2 both involves. When enzyme 1 is there, so it forms a red pigment, and if the enzyme 2 is there, then they modify the pigment into the purple. Okay. So what happens if both the gene capital R, capital P, capital T are there, then the flower color would be purple. If the enzyme 1, that means capital R is there, then the pigment color is red, right? And if the only capital P is there, then it is white only, right? Because red pigment is not there, right? Because if this gene is missing, then only the white flower would be there. So this is the red flower, capital R is there, right? So this is red flower. This is white. Both enzymes are absent. Here the first enzyme is absent, second is there, that's why this is white, right? And here both the enzymes are there, E1 and E2 both enzymes are there, so this is purple. So the ratio becomes, this is enzyme 1, enzyme 2, so this is purple, so 9 flower, right? This is red, so this is 3, and these two are the white flowers, so this is 3 plus 1, 4. So 9, 3, 4, right? 9:3:4 is the epistasis. But two different kind of epistasis are there actually. The one is dominant epistasis and another is recessive epistasis. The thing is that when the dominant gene, when the dominant gene acts only, right, then here you see the ratio. If both enzymes are there, then purple, right? And here if the recessive gene is there, this is the recessive, and this is the dominant one. So in the recessive epistasis, the ratio is 9:3:4, right? And in the dominant epistasis, if the enzyme 1 and enzyme 2 is there, then violet flower is there, right? If only capital V is there, again the violet flower is there. If the capital Y is there, then only yellow flower is there. Just to look at here, only Y is there, then it is yellow flower. If the both are independent actually, right? And here both are in the recessive form, so white flower is there. So this is 12:3:1. So this is the dominant epistasis. So here the non-allelic counterpart, right? Non-allelic counterpart is dominant here, right? In the dominant epistasis, right? And here the recessive in the non-allelic counterpart is dominant. Okay. So this is called epistatic gene, whichever is dominant, right? Whether it is recessive or dominant, but if it is finally responsible for making the character, then this is called epistatic gene, and another one is the hypostatic gene, right? So the ratio is like that, 5 minutes. Okay. Next is the duplicate gene. So duplicate gene is nothing but when enzyme 1 or enzyme 2, any one of them is there, then the color is red, the flower color is red, right? Any one enzyme, right? So see here, just we come to the inheritance only. So here R1, R2, that means enzyme 1, enzyme 2 is there, so red flower. So only one, so again red flower. So only R2, red flower. But here both are in the recessive form, so white flower is there, right? So altogether this is 15, right? And this is 1. So ratio becomes altered and it becomes 15:1, right? So Mendelian ratio here also altered. So this is called duplicate gene. And then this is additive gene. Next one is the additive gene. Additive gene, what happens? The solo action of two different genes or two different enzymes. Suppose enzyme 1, it makes a pigment, red pigment, which is 5 mg per petal, and enzyme 2 also makes a pigment which is 0.5 mg per petal, right? If both enzyme 1 and enzyme 2 are there, right? If both enzyme 1 and enzyme 2 are there, so this is dark red, right? And only one enzyme is there, this is light red. This is E2 is present here, so this is light red, and no enzyme, no E1, E2 is there, so this is white. So the ratio becomes 9:6:1, right? So this is 9, these two are 6 light, and this is 1, right? So this is the additive gene. Okay, so this is nothing but the summary of these genes, right? So this is the question, you just solve it. We come to the next, the polygenic inheritance. So polygenic inheritance is nothing but more than one genes are responsible for a single trait. So Nielsen and Ehle, they first time found in the kernel color of the wheat, and then Davenport, he worked on the human skin color and he gave a very interesting study. So this is the skin color of the human. It is controlled by the three different gene, A, B and C. If it is in the capital A, capital A, capital B, capital B, capital C, then this is very dark skin color. And if this is in the recessive form, then very light skin color. So if you make a Punnett square diagram, you will get this kind of ratios, and this ratio is 1:6:15:20:15:6:1. And if you plot this graph, you will get a bell shaped graph, this kind of graph, right? So mostly the tan color, right? Tan color is, most of the population, human population is tan skin color, right? So this is polygenic inheritance. More than one gene, it interacts for a single kind of character. So next is a complementation. This is very important and last topic of today's lecture. So complementation is nothing but when the mutations are there in the different gene. If suppose two genes are responsible for a single character and mutation are there in one gene, right? Then one can complement, right? In the hybrid or F1 generation, it can complemented by one another. Like here, capital A, small b, right? So the eye color is always white. Here also the eye color is white. This is small a, this is, but when you cross these two Drosophila melanogaster, then what has been observed, that eye color has been red here, right? The eye color is red. So this was explained by the complementation. See here, two different cases only I am discussing. So complementation only can be observed when the mutation are there on the different genes, right? So here the mutation is there on this A gene, and here the mutation is there in the B gene, right? And when you cross them, then you will get this kind of genotype, right? So here this green and this pink color, both will express themselves, and the wild type Drosophila has been observed from the mutant Drosophila, right? So what happens here, this A as well as the B, both are complementing to each other, right? But okay, so here the complementation occurs, right? So when the mutation are on the different gene, complementation occurs. Second case, when the mutation is there on the same gene, here A, and here also the mutation is there on the A gene, but at the different places. So it has been seen that Drosophila is again mutant here. So no wings are there in the Drosophila, right? That means this A gene is not complemented from anywhere. I mean, this is not supplemented from anywhere, this protein. So the mutant are wingless, right? So the complementation does not occur here, right? So mutation in the same gene but at the different location. So gene belongs to the same complementation group, right? So here, this is the last slide. So this star is the mutant, right? Wherever star is there, this shows mutant, and if dash is the wild type. So the question, by giving this table, anyone can ask you how many complementation groups are there. So here you can say the A and B, this gives one mutant, right? A and F, this is another mutant. So the complementation group, first complementation group can be made by A, B and F, right? A, B and F, they are in the same complementation group, right? Second complementation group can be made, BF already done, ye ho gaya hai, right? So C, here C and E, this is the another complementation group, see? So second complementation group. Jitna bhi aap dekhoge, all of them have that group only. C and E already kept into the second complementation group. Here also see B and F, first complementation group. Here A and F, first complementation group, right? So D here, if you look at D, there is no such mutant is there. So this can be kept onto the another or third complementation group. So here three complementation groups are there, right? So here another question is that for Drosophila, B and C mutation is in the same gene or in the different gene, right? So of course at the different gene, B and C, right? So B and C, they are not in the same complementation group, right? Same complementation group में नहीं है, right? See here B यहाँ पे है और C यहाँ पे है, right? So mutation is there on the different genes. So 2-3 minutes. Okay. So that's it. If you have any question, you can ask me. And now here I am ending my today's lecture. So thank you very much.
So thank you very much, Niraj sir. I am damn sure your concept about the classical Mendelian genetics is quite clear now, because he delivered so nicely, and even I have watched the lecture. And whatever questions you will attempt for any entrance examination, I am sure you will do that. So sir, thank you very much for sparing your valuable time with us, and for all these students. On behalf of GSBTM and NB Patel Science College, I thank you a lot, and thank you for sparing valuable time. Now, dear student, we will have 5-10 minutes break, and then next lecture will be taken by Dr. Rakti Mukherjee, that we will start within 5 to 10 minutes. Thank you very much. बढ़िया आपने को असल में देख रहा था बाहर एकदम क्लैरिटी आवाज में। आई थैंक्स दे दिया है, आ जाइए नीरज सर, इधर आ जाइए आराम से।
बंद हो गया।